Thank you for the kind introduction and the invitation to present in this session. I would also like to thank the participants for taking the time to attend this workshop considering the unusual circumstance we are all in. In the last session, the principles of method validation were discussed. In this session, we will go a little deeper. Specifically, Dr. Ahmed and I will discuss bioanalysis from a clinical pharmacology perspective. This presentation will be in the form of questions. I will initially pose a series of questions and will try to answer these questions during my presentation. So, what are the questions? First, why is bioanalysis important? I will discuss this question from a clinical pharmacology perspective. Second, what are the important questions that one needs to ask when developing and validating bioanalytical assays? 3. We'll provide an overview of what we as clinical pharmacology reviewers look at when evaluating bioanalytical assay. Next, we will discuss some of the bioanalytical issues we commonly observe, and I will provide some examples. Finally, I have a question for you. What can you do to help us? Usually, this is the question you often ask us as to how we can help you. Here, I'm turning the table, so to speak, and posing this question to you. So why is bioanalysis important? As a clinical pharmacologist, we evaluate clinical studies pertaining to the pharmacokinetics of the drug, the effects of intrinsic and extrinsic factors on exposure, and exposure-response relationship for safety and efficacy. These studies generate a bunch of exposure data that we analyze to make meaningful dosing decisions, not only for the indicated population, but also for the patient subpopulations, for example, patients with organ impairment or patients with concomitant medications. But what I want to impress upon you is the exposure results. These results are basically derived from concentration data, and that is where bioanalysis comes into the picture, and it forms the bedrock for the exposure data. Therefore, we need reliable bioanalytical assay or reliable bioanalytical assays to ensure the reliability of the concentration data. Therefore, we can make meaningful clinical decisions. So let's move to the next question, which is, what are the important considerations one needs to bear in mind when developing a bioanalytical assay? Notice that I said one needs to bear in mind and not one needs to know, because as bioanalysts, we all know, but do we always remember? So, I want to emphasize the following. The first question relates to assay purpose. Will it be used for studies to make dosing or labeling decisions, or will it be used in early development for go or no-go decisions? Once we know this, we know the expectation for the assay. The second question deals with specificity and selectivity, which is detecting signal from noise. The third question talks about the level of assay performance we need, which depends on the signal we are measuring and the resolution that we need to measure the signal. Once we know the allowable assay variability, we can then set the appropriate acceptance limits, which is the fourth question. The final question is, what can affect assay measurement during the study? So we need to know the expected sample handling conditions during the study. So these are some of the important considerations that should cross your mind when developing a bioanalytical quantitative assay. Before I discuss what we do, I want to give you a glimpse of the review timeline so that you have an idea of what we have to deal with within this timeline. When a new molecular entity comes in, it is assigned a standard review clock of 10 months or a priority review clock of 6 months. In areas like oncology, many of the new molecular entities fall under the priority timeline. These are some of the milestones during the review process. But what I want to impress upon you is the review timeline. This is 6 months for a standard review clock and only 4 months for a priority review clock. Now, as I mentioned earlier, as clinical pharmacology reviewers, we have to deal with a review of a number of clinical studies. Some of them are listed here. Bioanalysis forms a small part, nonetheless an important one. We realize this, and this is why we have an initiative now to scope bioanalytical package early in the review process for the completeness of the package. Talking about completeness, I want to emphasize the importance of a complete package. Incomplete information affects all of us, as it reduces review timeline and handicaps reviewers' evaluation. It puts the reviewers in an awkward position to make decisions with incomplete information, and sometimes this can have disastrous results, as listed in the slide here. What are the issues we look for when evaluating bioanalytical assays? We basically look for issues that could undermine the performance of the assays during validation and, more importantly, during in-study performance. Some of these issues are highlighted here. What we are basically looking at is the robustness of the assay and if there are any systemic issues, as these assays are often used in several clinical studies. We also pay close attention to complex assays. Specifically, we assess whether the handling conditions of the QCs reflect the study samples. So what do I mean by this? Let's say we have an assay that involves enzymatic digestion of study samples. So we pay close attention as to whether the QCs in these assays are handled in an identical manner as the study samples and how do they perform. Stability is another area that we closely look at. We want to know the robustness of the data. Does it adequately cover the study period? Are the stability QCs made with the right entity? Multiple methods. Cross-validation is another important area that we look at, because usually an assay evolves during clinical development, undergoes constant fine-tuning. At times the fine-tuning may be major, at times the platforms can change, and assays may be shared among several studies. So there is often a need for cross-validation during clinical development. So what are the issues that we often observe? Cross-validation is one, as we are often dealing with multiple assays that are involved in multiple studies. The other common issue is stability, and this often deals with incomplete stability information. The third area revolves around unexpected issues. These issues are usually glossed over, or their impact is not discussed in the analytical or the validation reports. The last two items revolves around the location and submission of validation and analytical reports. So these are some of the common issues, and some of them are critical and some of them are not. So in the next few slides, I'll go over some examples to illustrate some of these issues so that you have a fairly good understanding of where we are coming from. This example illustrates the need for prospective cross-validation with incurred samples. Cross-validation with quality controls is a necessary first step. However, cross-validation with incurred samples is also necessary. And this example tells you why. 4 iterations of the methods were developed at different stages of drug development. All the 4 methods were initially cross-validated with quality control samples. The method was initially developed and validated at Site 1. The method was then moved to Site 2 with some changes. Cross-validation with incurred samples indicated that the method at Site 2 was good for the drug, but not for the metabolite as indicated here. The applicant then validated a separate method for the metabolite and cross-validated against method 1, and the cross-validation passed as shown here. Similarly, at later stages of the drug development, the method was moved to site number 3. Cross-validation with incurred samples against method 1 showed that method 2 was good for the drug but not for the metabolite as indicated here. Therefore, the sponsor validated a separate method for the metabolite, and method 4 was found to be cross-validated as shown in this figure here. So the question is, are all the methods cross-validated to the original method number 1? Is this sufficient? If you play a closer look at the cross-validation with method 4, majority of the concentrations are near the lower end of the acceptance criteria for cross-validation. So it appears that there is a negative bias here. A comparison of the concentrations indicated that method 4 underestimates sample concentrations at high concentration levels compared to method 1. So is this an issue? This is not an issue in this case, as method 4 was analyzed only in a single standalone phase 1 study, and all the samples in the study were analyzed by method 4. However, this would have been a potential problem if the studies were cross-compared or analyzed by both methods, and also if the studies were used for dosing decisions or for labeling purposes. So this example talks about bias in cross-validation. Can we live with bias? Here, platform 1 and platform 2 were cross-validated as indicated in this figure. However, comparison of the subject concentration reveals that the platform 2 concentrations are higher compared to platform number 1. Also, we see that the platform 2 concentrations have a positive bias, but the bias appears to be constant. Correcting for the bias using mathematical transformations, one finds that the corrected platform 2 concentrations overlap with platform number 1 concentrations. Therefore, we can live with bias as long as it can be shown that the bias is constant. This example highlights the importance in continuously monitoring method accuracy during the drug development process. It's not sufficient just to validate the method and expect the method to perform well during study sample analysis, and this example illustrates why. Method 1 was validated and was used to analyze samples from a pivotal study and also from a supporting clinical study. However, during study sample analysis, the sponsors noticed inconsistencies in the drug concentrations. Therefore, the sponsor conducted an investigation, and the investigation showed that there was an assay interference that led to overestimation in the drug concentrations as shown in this figure to your right. Increasing interfering analyte concentration increases the drug concentrations as shown by the QCs here. So the sponsor, the applicant I should say, went back to the drawing board and developed a new assay and validated the assay and confirmed that there was no assay interference as indicated in the figure to your right. The applicant did not stop with this. They compared the concentration of a subset of study samples between the two methods and found that method 1 consistently overestimated the drug concentrations, confirming the results of the earlier investigation that we saw in the previous slide. So the applicant decided to reanalyze all samples from the pivotal study using method 2. However, for the supporting study, the study samples could not be analyzed due to sample integrity issues. But the applicant was able to demonstrate that the bias in method 1 was consistent and therefore was able to use a correction factor to correct for the overestimation observed with method 1. So these three examples illustrate the importance of using incurred samples for cross-validation and the comparison of concentrations between the methods. This example illustrates what happens when the bias is not constant. So we have two methods that were cross-validated. However, comparison of the individual PK concentrations reveals that there is concurrence at the early time points. However, the concurrence was not observed at the later time points as indicated here. So the bias is not constant in this case. And also, the exposure from platform 2 is about 30 to 40% lower than platform number 1. So considering the bias is not constant and considering a lower exposure with platform number 2, if the concentrations from platform 2 would have been used, then the patients would have resulted in a higher dose. This example addresses the issue of incomplete stability information. You may wonder how, but it does happen. Freezer conditions of study samples were altered midway during storage from a higher temperature storage condition to a lower temperature storage condition. Majority of the study samples were outside the validated stability period at the higher temperature storage condition. Only 30% of the study samples from the pivotal study were within the confirmed stability period at the higher temperature storage conditions. So, all in all, this is not good news, as there was insufficient study samples for PK modeling and simulation. This example addresses the issue of unexpected findings. As mentioned earlier, unexpected findings need to be reported and its impact addressed. Here, the applicant analyzed the drug and the metabolite in a phase 2 study. The analytical report stated that several samples were outside the validated stability period, but did not address its impact. An information request was issued to identify the samples and compare its concentration against the samples within the stability period. The applicant provided the information. The results showed that similar metabolite levels were observed for samples outside the stability period to those that were within the stability period. Also, these concentrations were similar to the phase 3 concentrations and were within the PK variability. So what is the conclusion? So this was not an issue in this case, as the metabolite had relatively low activity and all the conclusions were based on the parent drug. However, had the metabolite been important and was used for dosing decisions or labeling purposes, this issue would have gone through rigorous scrutiny to decide on the acceptability of the samples that were outside the stability period. So what can you do for us? Understand why you are validating an assay. Understand the purpose. Report unexpected results and address the impact of these results. Remember, incomplete information reduces review time, so make sure you have a complete package. One final thought. If you are developing a new technology or have assay-related issues, we highly recommend early interaction with the agency. The sponsors do this all the time for the other disciplines, but we rarely see bioanalytical issues being discussed. So, we want to make you aware of this option, and it is up to you to make full use of this option. So, if you have missed most of what I discussed earlier, make sure you remember this. Follow the FDA BMV guidance. I'd like to thank the following individuals, especially Dr. Brian Booth, for his invaluable input. Thank you for your attention and stay safe. So I was requested to provide 2 challenge questions to make sure that you've understood my presentation. I'm just kidding. So the first question is, you repeated long-term stability assessment as the first assessment failed due to a suspected conduct issue. Here the key word is suspected. So in the validation report, do you report just the long-term data from the second assessment, or you report long-term data from the second assessment and mention the reason for the second assessment, or you do number 2 and also provide data from the first assessment, or you do all of the 3, number 1, 2, and 3, and also provide pool data from both assessments. So I'll give you like 20 seconds. So the right answer is number 4, because as I said, the key word is suspected. That doesn't mean that you have found the problem. So you do not know whether there's a problem. And therefore, you have to provide all the data and you have to provide the pool data. from both assessments. Challenge question number 2. You decided to switch to a high throughput method during late clinical development of drug X. You have sufficient volumes of the study samples. You plan to cross-validate the method with blinded QCs and a subset of study samples, or blinded QCs and pooled study samples. So which is right? There's only one right answer. The right answer is blinded QCs and a subset of study samples. For those of you who went with blinded QCs and pooled study samples, you missed the part that you have sufficient volumes of the study samples. So when you have sufficient volumes of the study samples, then it's better to do the analysis with the individual study samples rather than pooled study samples. Thank you for your attention, and please hold on to your questions until the Q&A session after Dr. Hamid's talk is over. Thank you.